Good morning and afternoon, 2021 Smirchna's biology classes. You guys all got started. You're with Mrs. Huss today. I expect that we are on our top-notch behavior, helped her with attendance, and are dominating. You had a bell ringer over diffusion and osmosis, which we started talking about yesterday. That is the movement of particles in and out of cells, or water moving in and out of cells. We talked about how that happens across the phospholipid bilayer. Few of these questions are tough, but it will help you guys realize even when I've taught honors biology, you need to draw diagrams to help yourself out. And I'm not going to be able to do that perfectly on here, but I'll talk you through it and we'll get better as we go. So, number one, bless you, Simi. Which term is used to describe the carrying? You'll notice that's italicized. Things are always italicized on the ACT when they're important. Carrying of molecules across the membrane that does not require energy. Okay, so we know right off the bat here, since it does not require energy, it cannot be active transport. Since it does not require energy, it is not active transport. That one does require energy. Osmosis is the movement of water. This doesn't talk about water, so it's either B or D. And we know that the carrying of particles is helping it across. We know that another word for helping it is facilitating it. So, number one should be B, facilitated diffusion. Number two, what is the main difference between a carrier protein and a channel protein? If Mrs. Huss hasn't yet, make sure you guys please pause the video and watch the short little snip I left about a carrier protein versus a channel protein. What I would do really quick is just show you guys this picture right here. Like we talked about yesterday, a channel protein is where it just has one little groove that is specially designed to let certain things across. Whereas a carrier protein, like the video showed and this diagram shows, maybe even this one's better, it changes shape as it gets that particle to move across. So you'll see first the opening's over here, it lets it in, and then it lets it out, and it changes shape as it goes, just like we're kind of showing here as well. So that's really tough to maybe for me to have on your computer, but the main difference between a carrier protein and a channel protein, they're both types of facilitated diffusion, but a channel protein just has a little groove where a carrier protein changes shape to get things across the membrane. I'll have a short video here in a second, but there are certain protozoans, which are kind of like amoeba-like things, that squirt water out when they move. I'll even see if I can go pull it up here, but when they move, they kind of squirt water out of a little appendage they have on the end. But if water is getting squirt out of those amoebas, that means it must be pretty strong orange juice outside the cell because water is going to go dilute it. If water is going out of the body because it's more concentrated outside the cell, too much is hyper. So that would be a hypertonic environment where it's shooting water out because it's too strong on the outside. Okay, that's tough, but keep drawing pictures that help you. Go add the dots on the outside and show your arrow of water moving. Number four, osmosis is a process. Before I even go look at the answers, I know osmosis is water going from high to low. It's the diffusion of water going from high to low. So I'm looking for water. The only one that talks about water is letter D. Osmosis is a process that involves equalizing the concentration of particles, making the orange juice equal on both sides, not too strong, not too weak, by the movement of, there we have it, water molecules. Okay? Number five, I will take off if you are not drawing pictures on your test. This is just like the concentration gradients we drew in class yesterday. Draw a box on the left. Chamber A has 40% helium. Chamber B on the right has 20% helium. It says they're connected and that allows molecules to cross. So what will happen? We know that since helium is allowed to cross here, we'll go from high to low, which would mean we'd go from 40 to 20. Which means that helium particles will diffuse from chamber A to chamber B, which I believe is letter A. Number five should be letter A. I expect to see a picture with that arrow drawn, talking yourself through it. This is how we grow as problem solvers. On the back side, I got to kind of clean up my Microsoft Word document here. Number six says a hypotonic solution. So right now, everyone, if you haven't, go draw a circle. Outside of the circle, hypo means little. So outside the circle should be a few dots. Inside of the cell should be a lot of dots. And that should kind of help us. Hypo means little. So that means on the outside of the cell, it is less concentrated than inside. It is less concentrated. Hypo means less than. Number seven, What is the fluid mosaic model? This is old. We barely touched on this in our cell chapter, but we talked about how fluid means that it has to be able to move and be dynamic and not be too rigid so that it can move throughout objects and in our bodies. Mosaic, like those mosaic glass windows, are made up of many little pieces. As you guys will see in just a little bit here, the plasma membrane is made up of a lot of different pieces for sure. And then model just means it's a work in progress. So the fluid mosaic model, in short, everybody, is what we call the plasma membrane because it's made up of so many little parts that are moving and working together, and that is all working as one unit, so we call it a nice little model. But that is what we're talking about with this plasma membrane letting stuff off, letting stuff in and out of the cell. And you can see that fluid mosaic model right there. Number 8 talks about a specific question with diffusion. If we go look, this one says which one will reach trying to record a video. Equilibrium's where if we drop a red piece of food coloring, when will it be even throughout? So if I go look right away, I go cross out the 225 degree ones because I know the colder water will not move as fast and it'll take longer to diffuse. Now between 100 mils and 200 mils, 100 milliliters will easily diffuse much faster, just like the perfume. It'll fill a small room quicker than it will fill a big room. So that would be number 9 there. And you just need to talk about the ways that we can make diffusion happen faster, and that's with faster moving particles or having a smaller environment. Okay, so at this point right now, you guys should make sure your names are on those and pass those to the front. First block, pass them to Presley. Second block, pass them to Carson, and then you guys give them to Mrs. Huss. I will put those in the gradebook later. At this point, please pause the video. And if you could, Mrs. Huss, please pass out this worksheet that is called The Structure of a Plasma Membrane. You may pause it and then come back and unpause me in just a second. All right, I got Simi calmed down. Hopefully you guys all have this worksheet. So there's actually an other side. You're not going to start on this side, so everyone flip it over. I think some of the first questions are about a bathtub and your neighbor grilling. On the top part of that worksheet, you're going to do this first. You're going to discuss, is it diffusion or osmosis? Remember, diffusion is all kinds of little particles, whereas osmosis is always water moving. Then there's some vocabulary at the bottom. You will work with that with your notes with one partner. When you think you are done, you will take it up to Mrs. Huss. At that point, she will give you the cutout pieces that go with this, and this is on the back of your worksheet. Now, if I remember right, we did a cutout activity when I was gone in the hospital, and you guys kind of just said we looked at the key and cheated, and it was out, and that it is what it is, but we're not going to do that on this one. So this plasma membrane structure is something that I've let kids use on quizzes in the past, but you guys need to take it seriously. So on the left, you'll see here's our phospholipid bilayer, and then we have all these little structures in it, okay? So you guys need to look at them, and what you'll end up doing is using your Chromebooks and your textbooks if needed. And there's some things online that should help with this a lot, but we've talked about some of these things. We've talked about the phospholipid bilayer. The word glycoprotein and glycolipid look scary, but they just have both to do with glycogen, okay? Just like it says right here, they both have a short carbohydrate or a short carbohydrate chain. And then you need to look at which one of these looks more like a lipid and which one of these looks more like a protein and know what they do. A protein receptor site is where proteins go and land. And then we've talked about channel proteins and carrier proteins. So when you're all done, Mrs. Huss will have kind of the vocab words, and you'll cut out and paste what goes over here. And you'll cut out and paste what their role is and what they do in the cell. If it were me, I would just put a little bit of glue on or a little bit of tape, or maybe call Mrs. Huss over. She has the key, and I would hate for you guys to glue it all down and do it wrong. So this is something we need to take seriously. That is a hint. Maybe on Friday, if it's correct and you've used it right, maybe that will help you. But this is us going further into how things move in and out of cells, looking at the cell membrane and what it's made up of. And again, this really starts to help you guys understand the whole idea of the fluid mosaic model, why certain things are allowed in and certain things are allowed out. So do your best, take your time. After that, you guys will have a short little activity with osmosis, but give me your best effort. Do the other side of this worksheet first, check it with Mrs. Huss, then you guys will do this side. Go get them, do your best, help each other out, use your resources. See you later.